Dear students of TYBSc, we will start our next lecture number 7 on cell and molecular biology, particularly on ultrastructure of nucleus. Up to last lecture, we have seen that what is the nucleus, what are the nuclear membrane, and what are the nuclear pores. So now we will see in details about the nuclear plasm and nuclear material and the role of all the elements present in the nucleus. Actually, in nuclear plasm, DNA and RNA are the main elements and they play a key role in the cellular activities. As well as nucleolus is there in the center of nucleus, and we will see the actual working of nucleolus. So the ultrastructure of nucleus is quite as such that it has a double layered membrane which surrounds the nucleus. That membrane have a folds and called as endoplasmic reticulum. And endoplasmic reticulum is serially folded one over another. In between these endoplasmic reticulum, there are some intervals, and these are nothing but the nuclear pores through which the exchange of material to the outside of the nucleus is carried out. Or we can say nucleus and cytoplasm communicates through these pores regarding the exchange of information. So these are some irregularly shaped bodies are nothing but the chromatin material, that is DNA and RNA material are included here. In spite of this, the remaining material is nucleoplasm, or we can say the matrix inside the nucleus is generally known as nucleoplasm. In nucleus, there is a small nucleus, or we can say nucleus of nucleus is known as nucleolus, or we can say a central body of nucleus which controls the activities is called as nucleolus. So nuclear membrane is double folded and having inner and outer lamina or inner and outer folds, or we can say double membrane structure. So this is the basic arrangement of a nucleus. We will see in detail the ultrastructure and its role. And what are the materials present in the matrix of the nucleus. The enzymes of a nucleoplasm are mainly for the synthesis of the DNA and RNA. Means there are some enzymatic elements which play a key role in the formation of DNA and RNA, or we can say ribonucleic acid and deoxyribonucleic acid. In spite of enzymes, there are some minerals present in nucleus. These minerals like phosphorus, potassium, sodium, calcium and magnesium plays a key role in the physiology of the nucleus. So third material is chromatin fibers, nuclear membrane, then nuclear layer, nucleoplasm, then chromatin fibers. In nucleoplasm, a thread like chromatin fiber is present, and that can be observed by using basic stain in the cells. They occur during interphase stage. There are two types of chromatin fibers present in the nucleoplasm, that is heterochromatin and euchromatin. Heterochromatin are darkly stained material or bodies present in the nucleoplasm that occurs around the nucleus, that occurs around the nucleolus of the nucleus, and it contains more RNA than DNA. And euchromatin is the another body or fibers that is highly stained and contains large amount of DNA in the cells. Means heterochromatin is RNA containing body in short, and euchromatin is DNA containing bodies present in the nucleoplasm. Nucleolus, this is acidophilic body present in the cell. It is absent in bacteria and yeast cells. The synthetic activity depends on the size of the nucleolus. The nucleolus is small in sperm or blastomeres or muscle cells, etc. And mostly it contains the RNA bodies, means single-stranded material, or we can say single-stranded chromatin material. So functions of nucleus. The role of nucleus is generally the controlling mechanism. So what it controls? First, it controls the all metabolic activities. Then nuclear membrane allows the exchange of ions from the cytoplasm. Nuclear membrane acts as barrier for potassium, sodium and chloride ions. The nuclear pore allows to move mRNA. This is basically for the protein synthesis. This is maintaining the potentiality of the ions, and it controls the metabolic activity, means digestion of the cell or energy release of the cells, and likewise activities. The nucleus controls inheritance of characters. As the nucleus have RNA and DNA, so DNA is the genetic material and RNA are the materials which are cooperating, helping, or we say preparing the protein molecules. And nucleus at last, we see here that it performs the function of biogenesis of ribosomes, because ribosomes are the materials which are cooperating in presence of RNA for the processing or for the production of protein material, or we can say mRNA and ribosomes play a key role in protein synthesis. And in this way there are many functions performed by nucleus to maintain the physiology of the animal body. Next one is the ultrastructure and functions of endoplasmic reticulum. So we have seen first of all the plasma membrane, then nucleus, then nucleolus, and now ultrastructure and functions of endoplasmic reticulum, or ER. The cytoplasmic matrix of the cell is traversed by a complex network of interconnecting membrane wound vesicles. These are called as endoplasmic reticulum. If you would like to see, you can see in the previous picture that this one is the endoplasmic reticulum. Here is the title. This nuclear membrane having a fold, and these folds are nothing but the endoplasmic reticulum. This might be another fold, then third, fourth fold. So in this way there are so many folds, endoplasmic reticulum bifold communicating to the cell membrane. So these are the endoplasmic reticulum materials. These are of two types, rough and smooth. We will see in details. The cytoplasmic matrix of the cell is transversed or traversed by a complex network of interconnecting membrane bound vesicles, and these are called as endoplasmic reticulum. These are often remain concentrated in the endoplasmic region of the cytoplasm and therefore called as endoplasmic reticulum. The name derived from the fact that in the light microscopy it looks like a net in the cytoplasm. It is looking like a net in the cytoplasm. It was first observed by Porter in 1945 under electron microscope from the liver cells and gave the name ER, or endoplasmic reticulum, in 1953. Later on, Fawcett, Ito and Thierry in 1958 and Rose and Pomeroy in 1960 have made various important contributions in the study of endoplasmic reticulum and observed in the laboratory and described the structure and its morphology, and they reported that The endoplasmic reticulum may occur in following three forms or different forms, each bounded by a unit membrane made up of protein and lipid molecules. First is cisternae. So these strands, one over the another strands, are nothing but the cisternae. If they form apices in a globular manner, we can say that they form the vesicles. So likewise, the structure of Golgi complex, we can say. So cisternae these are long, flattened, sac-like, unbranched tubules having the diameter of 40 to 50 micron. They remain arranged parallel in bundles or stakes. If we keep the coins one over another, it looks like a endoplasmic reticulum, cisternae of the ear, stakes one over another. And if they have attached ribosomes to the cisternae, these are called as rough endoplasmic reticulum, RER, rough endoplasmic reticulum. And usually exist as a cisternae which occur in those cells which have synthetic roles as the cells of pancreas, liver and brain. So these cells have stranded cisternae which are stakes or stacked one over another. So these are the cisternae. You can say 1, 2, 3, 4, and these dots are nothing but the ribosomes which are attached on the cisternae. So this is nothing but the endoplasmic reticulum. And if it is pinches of a small globular material, it will be called as vesicles. So these are the vesicles. And if they have, instead of forming such a large globular structure, if they are tubular or branched tubular or in this way, so these are known as tubules. So these are the different components of endoplasmic reticulum. So we have seen first cisternae. Now second we will see vesicles. Vesicles are oval. These are oval, membrane bound vacuolar structure having the diameter ranging from 25 to 500 micron, and they often remain isolated in the cytoplasm and occur in most cells, especially abundant in smooth endoplasmic reticulum, means SER. If we remove all the granules or ribosomes, then it will be smooth endoplasmic reticulum. And these dotted or ribosome containing cisternae are called as rough endoplasmic reticulum. So this is the difference between RER, means rough endoplasmic reticulum, and SER, smooth endoplasmic reticulum. So this is about cisternae and vesicles. Now we will see about tubules. So these are branched structures forming the reticular system along with the cisternae and vesicles. Along with cisternae and vesicles, some strands are also, tubular strands are also found, and they are called as tubules generally. Actually these are endoplasmic reticulum material. They usually have the diameter of 50 to 190 micron and occurs in almost all cells. They occur almost in all cells. Tubules will found in generally all cells. Now, the type of endoplasmic reticulum. As I told you previously that if they are attached with granules, it is called as granular, and if they have no granules, it is smooth or agranular. So granular or rough endoplasmic reticulum and agranular or smooth endoplasmic reticulum, SER and RER. We have discussed it previously, but now we will see in detail the structure. This type of endoplasmic reticulum, that is agranular or smooth, possesses smooth walls because the ribosomes are not attached with its membrane. And this type of endoplasmic reticulum occurs mostly in those cells which are involved in the metabolism of lipids and glycogen. Example, adipose cells or interstitial cells or glycogen storing cells of the liver, conduction fibers of the heart, spermatocytes and leukocytes also containing this type of smooth endoplasmic reticulum. And although the smooth endoplasmic reticulum form a continuous system with a rough endoplasmic reticulum, so they are very closely related with each other. If they have granules, it is called as rough endoplasm. If they have no granules, they are smooth endoplasm. It's very simple and maybe continue in association with each other. Now, B part is a granular or a rough endoplasmic reticulum. We can call it as RER. This type of endoplasmic reticulum possesses rough walls because the ribosomes remain attached with its membranes. And ribosomes play a vital role in the process of protein synthesis. As I told you previously that mRNA and ribosomes play a key role in the formation of proteins, or we can say protein synthesis. Example, pancreatic cells, plasma cells, goblet cells and liver cells. These are the major types of cells. So functions of endoplasmic reticulum, as previously we have discussed that the endoplasmic reticulum acts as secretory material, therefore they form vesicular sacs. Then storage, then circulatory and nervous system for the cell. It performs various following functions. First, mechanical support. Endoplasmic reticulum gives mechanical support to the cell by giving its strands or folds. The ER divides the fluid content of the cells into smaller components and thus provides mechanical support for the colloidal cytoplasm. So this is the first and important function, that it supports the mechanical strength to the cell. Second, the protein synthesis. As previously I told you that the protein synthesis is being carried out in presence of ribosomes and endoplasmic reticulum. Actually, ribosomes are the granules. They, in presence of RNA and some amino groups, form the protein molecule. Therefore, the rough endoplasmic reticulum is the site of protein synthesis, where actual protein molecules are formed, and the proteins are synthesized on the ribosomes and enter the endoplasmic reticulum cisternally through the channels in the membrane, that is secretory proteins leave the endoplasmic reticulum and enter the Golgi complex, and from where they are secreted outside the cell in the form of vesicles or sacs. So in this way, the protein synthesis is the main activity carried out or facilitated by it. Third, glycogen synthesis and storage. These both activities are carried out. In fasted animals, the residual glycogen has been found associated with the endoplasmic reticulum. So one of the main function of ER is during fasting, whenever the food is not there in the body, the glycogen synthesis has been occurred. Fourth function is lipid synthesis and storage. As the protein is synthesized, lipid molecules are also synthesized and stored in the body with the help of ER. In association with, we can say, synthesis of triglyceride, means 3 molecules of the glyceride, as well as intracellular transport and storage of lipid material is occurred. So very simple. Fifth function is synthesis of cholesterol and steroid hormones. Once it is forming lipid and protein, naturally the synthesis of cholesterol and steroid hormones are there. Because all hormones are steroidal or fat material containing nature. So ER is the major site for cholesterol synthesis. SER is believed to be connected or concerned with both the synthesis and storage of cholesterol. The testes, ovary, adrenal cortex, etc. The cell shows SER, smooth endoplasmic reticulum, and it has a role in the synthesis of steroid hormone. So steroid hormones in the body are mainly synthesized by smooth endoplasmic reticulum in the cells, specifically in the testis cells, ovary cells, and adrenal cortex gland cells. Cholesterol is an important precursor of steroid hormone. So it is a precursor of steroid hormone. Therefore, cholesterol is one of the important fatty material saturated in the body. Sixth function is detoxification. The smooth endoplasmic reticulum contains an enzyme, and that enzyme system is concerned with detoxification properties and controls the toxicity material or acidic material of the cells. In this way, endoplasmic reticulum performs many functions. So before to study the next one, I would like to inform you that the seventh function is circulation and exchange of material. The endoplasmic reticulum acts as an intracellular transport system for various substances. Watson in 1959 suggested that the exchange between the nucleus and cytoplasm takes place through the nuclear openings and which communicate with the endoplasmic reticulum. So this was suggested by Watson during 1959. Then substances may pass across the endoplasmic membrane or endoplasmic reticulum membrane by diffusion, simply by diffusion, as well as by the active transport. So both ways shows the transport of material or substances. Eighth function is membrane flow. The transport of ions, molecules, and particles into and out of the cell may also take place through membrane flow. For example, the substances like RNA and the nuclear proteins may pass out from the nucleus to outside by following the route nuclear membrane. Then it passes to the nuclear pore. From nuclear pore, it passes to the endoplasmic reticulum. From endoplasmic reticulum, it passes to the Golgi complex or Golgi bodies. From here, it passes to the plasma membrane, and from plasma membrane, it is thrown outside the cell body. So this is the way through which the membrane flow is being carried out, or membrane flow system is maintained or regulated. Ninth function, or we can say it's the last function. In spite of that, many minor other functions may be carried out, but these are the major 9 important functions. It includes other synthetic functions, that is, smooth endoplasmic reticulum synthesizes the steroid compounds like triglycerides, hormones, testosterone, and progestin. Means these are the hormones, and maximum many type of hormones are either in protein or fat material, and these are synthesized by the smooth endoplasmic reticulum. In spite of that, it also synthesizes certain visual pigments from vitamin A. So this one is the peculiar function and rare function, that it synthesizes certain visual pigments from vitamin A. So here is the functions of Yeah, so we will stop the discussion here. So we will continue our further our next lecture, in which we will study the ultrastructure and function of Golgi bodies. So next molecule will be studied in the next lecture. Till then, see you.